Okay, so let's start. Good morning. So, in this course, we are towards end. We are discussing some advanced topic in fluorescence spectroscopy. And if you remember, last class we had discussed some of those advanced application of fluorescence. So, we started looking at the fluorescence polarization. We looked at fluorescence anisotropy and lifetime measurement, and how you can use this concept of fluorescence polarization or lifetime measurement to understand the dynamics of the protein or fluorophore and what is surrounding that. We looked at the application of this lifetime measurement or anisotropy in finding it out antigen antibody interaction. We also looked at how association and dissociation of protein happens, and you can study using these fluorescent based concept. And even protein folding, like a helix to coil transition, this is one of the important aspects in protein recognition, how helix to coil transition happens. You can study using fluorescence concept like a lifetime or polarization and all those, right. Do you know where helix to coil transition plays an important role before I embark upon today's concept? Any idea about helix to coil transition? Is it important or not important? Important? Very good. Signal transduction. So, you know, human, like a proteome or eukaryotic proteome, about 1/3 proteins are unstructured. They are called intrinsically disordered proteins or intrinsically disordered region. They do not have persistent structure, but they can adopt the structure depending upon what is around, right. Context dependent structure adoption they do, they adopt their structure, it depends upon what is coming. So, for suppose this is my fingers are IDP, intrinsically disordered protein. If it has to bind to this chalk, it will come like this. If it has to bind to this bigger one, it should be coming like this. So, this is a coil transition, it can take another conformation, one of them is helix conformation. Where it is going to be used, that is the concept you can study, the transition between helix and coil using fluorescence anisotropy or polarization. Now, today we are going to discuss one of the important concept that I was already telling you. It is called fluorescence resonance energy transfer, FRET, right. So, FRET essentially it is a fluorescence based spectroscopic technique to measure the distance between two fluorophore, fluorophore 1 and fluorophore 2. And you know, when we are measuring the distance between two fluorophore, now this distance can change, right, because protein have motion, they have a breathing motion, they are folding, they are doing something like this, they are doing something like this. So, if I can measure the distance between two fluorophore, the distance is changing, right. Therefore, I can understand not only the dynamics associated with one fluorophore, but how they are correlated between like these two fluorophore, and that is what FRET enables. But to happen FRET, you require two fluorophore, right, that is the primary requirement. And another important requirement, one will be donor, another will be acceptor. Donor will give the energy, acceptor will accept the energy, and that is the concept of fluorescence resonance energy transfer. So, energy is getting transferred from donor to the acceptor. So, energy transfer is happening. Fluorescence is clear. What is resonance? There is a term called resonance. What is resonance? Electron shifting? No, electron shifting is not resonance. In simple term, what resonance means? Simple term में बताओ. Matching of like a frequency. Matching of frequency. Your friend is resonating with you. You are resonating with environment. That means your frequency matching with other frequency. Two people resonating each other means the if I am resonating with her, my thoughts, my frequency is matching with her. That's we are in sync in terms of frequency, right, synchronized in terms of frequency. So, here also the frequency match has to happen between a donor and an acceptor, and if this frequency match happens, energy gets transferred from donor to acceptor, and that is what we measure it, right. So, primary condition for FRET to happen, it is a two fluorophore system. So, one is donor, one is acceptor, and it has to be in close proximity because energy has to be transferred. Close proximity means like a 10 to 100 angstrom distance they are. The another is absorption spectrum of one should be matching with the emission spectrum of the other, right. So, if I just go on the board, say this is my spectrum for a fluorophore, here is absorption and this is emission, absorption, this is emission. Now, it says that the emission of this guy should match with the absorption of the another fluorophore. So, here is absorption 2, emission 1, and then there will be emission of that guy, right. So, it says that emission of first molecule which is donor should be matching with absorption of second molecule which is acceptor. And the third condition, donor and acceptor transition orientation must be parallel. That means it is a EM wave, right, based radiation. So, transition dipole should be like this. If it is like this, they will not be efficiently transferring. So, this is the third condition. Second condition, absorption. Emission condition, the emission of donor should be matching with absorption of acceptor, and energy has to they should be close. So, essentially this is the donor ka fluorescence, acceptor ka fluorescence. Donor emission should be matching with, and then only energy transfer can happen, right. So, this is a critical requirement. You can put these two probe anywhere in the protein, right, that design you can do. How do you put donor and acceptor in a protein? These are, so if tryptophan is say donor, it is already there in the protein, but you have to find an acceptor, right. If you want to put donor acceptor both to be put as extrinsic fluorophore, you can put it by doing some mutation. Where do you tag? You want to tag a fluorophore, where do you tag?
In the protein sequence.
No, we are working in the, the extrinsic fluorophore cannot be coated in the protein sequence. Whatever you are saying, GFP is like a GFP, YFP, RFP, but that is a whole protein you are coating. I am just talking about a small fluorophore. If I am doing experiment with a 10 kDa protein, I cannot coat 30 kDa protein or 40 kDa protein which is donor or acceptor. So, I want 500 Dalton or 1000 Dalton fluorophore. So, essentially you can design, or you can like someone can design a fluorophore which is small enough, has satisfying this condition, like a emission of a donor matching with absorption of a spectrum, and you can tag in the protein by covalent linkage. And how do you do that? Create a cysteine mutant in the protein, and you can form the SS bond where covalently your acceptor or donor can be tagged. So, because of this chemistry, the people have come up with several of donor and acceptor pair, like just pay a little bit of attention to that. It depends upon where to where you are to transfer it, right. First it is small molecule based, then whatever he was talking, the protein based. So, if you are looking at the violet color, the argon, like a laser you can use it, then it is a donor is Alexa fluoro 405, and then acceptor should be Alexa fluoro 430. So, excitation donor excitation and acceptor emission is 405 you excite, and 531 finally you absorb the emission. Similarly, again with argon laser you are psi 2, it is a donor, acceptor is psi 3, these are the wavelength 488 and 566. Then psi 3, psi 5, these are commonly used donor acceptor pair. Then FITC trick 488, 577, P APC 488 660, then Alexa series of Alexa 488 488, you can have a Alexa fluoro 514 532 and all those. So these are typically a donor acceptor pair for a small molecule. You can have a fluorescent protein, that if suppose I want to do not in the isolated protein system, but I want to measure the protein inside the cell, right, then it will be difficult. So whatever he said, put a reporter gene, the protein which is expressing GFP, you can have a CFP and can have a acceptor pair as a YFP, yellow fluorescent protein. You have a purulene fluorescent protein, yellow fluorescent protein, GFP, YFP, GFP, MRFP, like a red fluorescent protein. So these are typical donor acceptor pair that people have researched and found it. The concept is again extremely simple. Here how all of them follow the same route. I just want to say I use 4 chalks to have in the 4 color. So let us see, we are exciting donor. So excitation of donor, right, where the emission will come, it will be shifted. So it will be emission, excitation of this emission of donor. So if you look at here, donor, excitation and emission we are writing. So this is our emission of donor, right. Now what, according to the concept, what has to happen? If my here excitation of acceptor is happening here, it will not give fret, because now it is not overlapping. For fret to happen, this guy has to be here, right, and this is the overlap that we are talking about. Overlap, this is happening. So what I have in yellow, it is a excitation of acceptor, and then we have finally emission of acceptor, right. So you excite directly here, you are exciting directly here, and you are absorbing emission directly here. So this is the excitation, 405 you are exciting for the first pair, and you are getting 541 as a emission. So this is the primary condition, you excite only at donor and you absorb emission at the acceptor, and this emission is only coming if they are in close proximity, they are talking to each other, they are in close proximity. So donor ka energy is getting transferred to the acceptor, and that is how it is getting excited and finally emitting. This only can happen if they are in close proximity. If they have overlap integral, like that is the that's the resonance condition we are talking about, and if this happens, you can get the energy transfer, right. So how much energy can get transfer? So the primary condition is this: donor, acceptor, emission, excitation has to match. Emission of donor should match with excitation of acceptor, then only energy transfer happens. They should be in close proximity, and their dipole should be like this. If it is this or this or this, they will cancel each other, so not energy transfer happen. The another important thing is your distance, and that is called the Foster distance. So energy transfer efficiency depends upon the inverse 6th power of the intermolecular separation. So how close or how far they are in the distance, that gives you efficiency of the transfer. R0 to the power 6 divided by R0 to the power 6 plus R into 6. R is a Foster radius, that is a fluorophore dependent phenomena. The R is distance between donor and acceptor, and E is the transfer efficiency. So even if your donor and acceptor, right, even if donor and acceptors are a very good fluorophore, if the energy is not matching, like the transfer is not happening, it will not happen. If they are too far, not happen. So this distance R is extremely important, right. and R0 is Forster distance is also playing role. So these are the two vectors, right? Two fluorophore have two vectors. The distance between them dictates the efficiency of that. So if you look at this curve, y axis energy transfer, x axis R by R0, if it decays with a time and 50% efficiency where R becomes R0. So the separation between two fluorophore should be equal to the Forster distance, that is a 50% efficiency. So that gives the efficiency of a fluorophore, right. So yeah, so now looking at the efficiency, so if you are doing this experiment, essentially you can measure the distance between donor and acceptor in a time dependent even manner, right. Time dependent manner you can measure it. So time dependent manner you can measure, that means we are looking at how they are closing, opening, how closing and opening, that is what you can do it. So the distance at which 50% efficiency happens of the excited donor and deactivated are deactivated by FRET is defined as a Forster distance. The magnitude of R0, which is Forster distance, depend upon the spectral property of donor and acceptor dye, what are these. So it is a dye dependent phenomena, and the rate of energy transfer is given by the formula KT 1 by TD, that is a TD is decay rate of the, like whatever we have discussed, the decay rate of the donor, R is Forster distance, smaller r is a distance between donor and acceptor, and rate of energy transfer is given by KT. So one can get it even the transfer efficiency, the distance between the donor and acceptor, and even the orientation vector between these. So you can see now by doing this FRET based experiment, we are able to measure the distance between two fluorophore in the protein, in a protein, right. Just what we are doing, so we are measuring the distance between two fluorophore. Suppose I want to get a complete map of the protein, what I need to do? Fluorescence based experiment you have to design, and you find a complete distance between these, complete distance map of a protein. So let us see, I have this protein and I do not know much about this protein, I know that some loopy structure it is. So now we have a sequence. So one way in today's era you put the sequence in alpha fold, you can predict the structure. But remember, alpha fold is a prediction, it is not experimentally validated. Nowadays since prediction power has gone up, so one can rely on that. But suppose you have to validate that structure, so what you are going to do, you put the FRET pair at different location, something like this. You can keep putting the FRET pairs and you are keep measuring the distance between these FRET pairs, like you just creating mutation, various mutation where we are tagging one fluorophore at a time. So two fluorophore, one donor, one acceptor, then change the position, one donor, one acceptor, one donor, one acceptor, and you created various FRET pairs in the protein. Now you did the FRET, measure the distance. So by measuring that distance, we measure the distance between this amino acid, that, all these distance you measure, and by measuring all such distance you can create a map of the protein structure. So essentially using the FRET based experiment you can create the distance map, right. That is one of the nice things about FRET. So where do you apply this FRET? Of course, in understanding the protein folding, the co-localization assay in the cellular context. If your protein A is co-localizing with protein B and they are giving or communicating in the cell, you can find it out. If they are coming together, they will show the FRET. Otherwise, if they do not come together, they will not show the FRET. So co-localization assay. You can develop again a biochemical sensor. You can do even nucleic acid based hybridization. So you tag one fluorophore on one strand, another on another strand. If they come closer, they will show FRET. And even membrane dynamics you can put it. So this guy in membrane, you know, it is a fluid structure. So put a fluorophore on one system, another on another system, and see how they are diffusing in the membrane, right. So, they are diffusing, when they come closer they show fret, and when they go, the fret efficiency drops or they will stop showing. So, all sorts of dynamics one can find it out using fret. I am giving you a very nice example in coming few minutes of understanding the dynamics of a protein in a real complex system. Are you clear? Okay. Any other question?
Sir, it will not be fixed on the base, right? It will be like fluctuating here and there.
Protein fluctuates here and there.
So how will we like create an image as such? If we are having one acceptor and one donor, it will be really difficult for us to observe in a medium where these two might be.
So that's why. So there are two things. So first, always you get an ensemble distance between them, right? If you do just fret, you can do time resolved fret, right? So time resolved fret, suppose this is my donor and acceptor, and this kind of motion is happening. So if you are doing say just fret, I get an average distance between these two. If I do time resolved fret, remember last class we discussed time resolved experiment. If you do time resolved fret, you can get a distance when they come closer and go far. So that can give even your dynamic picture of a protein, how closely its closage open, this distance is changing, that you can measure it. The only condition, it has to happen in nanosecond time scale. If it is happening slow, you get a static picture. If it is happening too fast, you cannot capture. So time scale of the fluorescence and your protein motion should be in the same range, then you can capture this event dynamics happening in the protein. Okay, so let us move it. Yeah, so proximity of tryptophan in an active site you can study. The spacing between hydrophobic group in bovine serum, something like that, protein dynamics. Determination the shape of visual pigment in rhodopsin. Rhodopsin, you know, right? All of us are seeing because of that. You can get the shape of this pigment. You can get the conformational change that is happening on substrate binding. Say this is doing motion like this. That substrate upon it's open, and here is my substrate coming, closing it. You can get this motion of the substrate binding using fret kind of experiment. The bigger even people have done the structure of ribosome, yeast, phenyl tRNA, all of such things you can get it. Let me show you two interesting example of application of fret. Here I want to combine two experiment. One is NMR based experiment, another is fret based experiment. Little bit background of NMR, this is not an NMR course, but I will tell you. What in NMR, what you detect? Nucleus, nuclear magnetic spin, and because of that you get an picture of a protein. So here in NMR there is a phenomena called paramagnetic relaxation enhancement, PRE, paramagnetic relaxation enhancement, and this relaxation enhancement happens if there is an unpaired electron. What it does? It enhances the relaxation of a nuclei. So PRE experiment, we do it in NMR to measure the distance. So in this experiment, what people have done, they put an radical using the same cysteine concept, NO radical, nitroxyl radical, which is a unpaired electron, right? Now this protein, and here is some other nuclei that we are detecting it, right? So when it is far, no relaxation, like no effect of this paramagnetic relaxation enhancement. When it come closer, you see that like a nuclei from here are started vanishing. So this has a distance dependent effect. So 10 angstrom suppose. So all those you see here in red are showing that nitroxyl radical is sitting here and enhancing the relaxation of these nuclei, and therefore you see the effect in this open state. When this comes in closed state, you see also many of the nuclei are vanishing from here. So it says that this opening and closing behavior happening, and therefore many nuclei which otherwise are nicely looking at, now they start relaxing fast, and you can see the closing and opening behavior of this protein. When nitroxyl comes ready here, nuclei from here gets affected. When it goes far, it does not happen. Okay? The same thing was done using FRET, fluorescence resonance energy transfer. I put a red dye here and a green dye here, right? Now they are FRET pair. From the list we chose that. And you remember what I am doing, here is a bond formed, right? So we are putting with covalent bond, otherwise ये भटकता रहेगा इधर उधर protein में. So I have fixed it, that I know the location where my red dye is fixed. I chose a location where my green dye is fixed. Now when they come closer, they will show FRET distance. When they come closer, the distance reduces to 36 angstrom, otherwise it is a 46 angstrom. So here you can see closed state and here you can see open state. So open state versus closed state, you can see this is again time resolved FRET we are doing. So you can find the fluctuation that is happening and you can fit it to understand the distance between open state and closed state. It is a reducing by 10 angstrom, and doing this time resolved FRET you can measure the distance between the open state and closed state, and when it is some intermediate state as well. So you are now using this FRET experiment able to see the motion that is happening in a protein. And these two experiments were done, NMR based PRE experiment, paramagnetic relaxation enhancement, and fluorescence based FRET experiment to understand the opening and closing of protein, which is say enzyme doing this breathing motion or opening closing motion. That is what one of the application in the protein. So now you can visualizing how protein is doing its activity, right? Otherwise if you take just a static picture, I do not know whether it is in closed state or open state, but here we can really capture that. Let us go to another example. Are you aware of this system? मेटाबॉलिज्म में पढ़ा होगा, एनर्जी एटीपी कैसे बनती है, राइट? All of you have studied in that metabolism course. This guy is sitting in the membrane. It is called F0, F1 ATP synthase. How it works? It's a molecular machine. How it works? इसका काम है ATP बनाना. How it makes ATP? ADP and PI attach and ATP happens. To work this machine, you have a proton gradient. This goes from down, like from intracellular to extracellular. That basically helps in rotating this motor, and then this motor rotation puts the ADP PI and makes the ATP. That is what we know, right? Electrochemical energy to a chemical energy. Where it works in our body? Mitochondria. Mitochondria में proton gradient कहाँ से आता है? So you have a glycolysis, then you have a TCA cycle. TCA cycle say it goes to electron transport chain. At the end it creates the proton gradient. Proton gradient is used for rotating this motion. How do you know that motor rotates? How do you know? It's a textbook. Your textbook is flooded with such information. How do you know as experimentalist that your motor rotates and rotates with certain speed? FRET करना पड़ेगा, राइट? ऐसे FRET ने बताया है कि मोटर रोटेट करता है. So you require such kind of a probe which shows that motor rotates. You can design an experiment. This is called single molecular FRET to understand that it happens. Single molecule FRET, right? If you take it bulk, कभी कोई मोटर ऐसे रोटेट कर रहा है, कोई शांत बैठा है, कुछ और किया है, right? So that will not give you the nice picture. So you have to go down that you can detect each of this molecular machine, and that technique is called single molecule FRET. So you have to really be very, very patient. Go to you have to keep all your nerves under control. रात भर लैब में अंधेरे में बैठ के एक मोटर को देखना होगा. Then only you can get it, right? So this is single molecular fluorescence and FRET for site directed motion. Site directed motion means you have to put somewhere a fluorophore so that when motor rotates it goes like this, right, and you can measure that in a time resolved manner. So that is what was done here. Two fluorophore were kept at this shaft, I would say, red and green, and when this shaft is rotating like this, the distance between them is changing, and you can plot it in a nice confocal volume. Here when they are rotating, you get it. So ATP hydrolysis and ATP synthesis that is happening, you see FD, fluorescence of donor versus fluorescence of acceptor at 2-3 levels, and fitting all those ratio, you know that motor is rotating between 1 to 2 to 3, and you get a complete picture of motor rotation, right. So this kind of experiment people have done it. If you are interested, so 10-20 years back these experiments were done. That is the data that came out. Okay. The another topic that I want to touch upon, if you are interested, you can later on study in detail. Can we use this fluctuation that happens in the fluorescence to understand something? Fluorescence fluctuation, you know, like suppose I take this a small volume and I detect only that volume. So now in solution this molecule are not static, a small volume, picoliter, femtoliter, whatever. So molecule enters into this focal volume, goes, enters, goes, enter, goes. So a small fluctuation they will show. So this molecular mechanism that can happen because of this fluorescence fluctuation can comprise the particle movement that is happening, the conformational change that happens, and any photophysical or chemical reaction that happens. So essentially if you look at, I am measuring in a small volume, molecule enters, going, even if they are doing reaction, we are measuring this. And this experiment is called, like this experiment of small focal volume measurement that we are measuring is called FCS, fluorescence correlation spectroscopy, FCS. That is what we are doing. So this is the setup. You have a sample and a cover slip. Now sample is sitting on the top, you have a laser and lens and dichroic mirror, emission filter lens and all those, and APD for detecting it, computer to processing it. So fluorescence emission of 2 dye at appropriate filter, you can get the correlation setup, right. You see the fluorescence of green dye and red dye, and we have same fluorescence based experiment, but we are now doing in a extremely small volume with precise one, but here you require a laser, right, laser to excite it. For an example, I give you, say, just one thing I just want to, what keeps you happy, which hormones or neurotransmitter or something keeps you happy? Dopamine, very good. Anything else? Serotonin, all these, right. Can I measure in the brain of a person if serotonin level is going down or up? Can I do that? If you are happy, I know that serotonin is okay, right, but can I experimentally measure it? Serotonin cell में आ रहा है भी या नहीं? Such kind of experiment if I am thinking, so I cannot do in a human brain or human tissue. It's a difficult and ethically not possible. But what about trying to do such experiment in mice brain? If I take the ethical clearance and sacrifice one mice, right, to understand the dopamine concentration, dopamine fluctuation, serotonin fluctuation and all those, you can really know that what is happening, right. And if in disease condition, आजकल बहुत लोग को डिप्रेशन होता है, डिप्रेशन में क्या सेरोटोनिन रियली गायब हो गया है या किसी और स्टेट में चला गया? Can I do such kind of experiment? So for doing such kind of experiment, you can put this kind of the setup where you have a sample cover slip in some tissue or cells, fine slice. Now it is not a solution, I am putting it in a cover slip, in a sample sector and covering it, and you can do this kind of experiment, and fluctuation that in a small volume that is happening, one can measure by such experiments, right. So here is a sample volume, you see that small observation we are happening from here, it is a coming, going, coming, going, coming, going. And now what we are measuring? The fluctuation of the intensity with time. So we are measuring intensity at time t and intensity at time t plus tau, and we plot something called correlation function. G of t is a correlation function with respect to time, and then we can measure this tau d decay of that. So one can do it fluorescence correlation spectroscopy by measuring the fluctuation in this small volume, and we can see how much concentration happening, how much concentration is going, and all those. This is simple some equation you have that essentially we are measuring the G of t, the change in the fluorescence at time t correlated with a change of fluorescence at t plus tau, and averaging over the fluorescence. So if you do here, you can plot an autocorrelation function, and you can see how the overlap integral is changing as they come and as they go. That same similar concept you have, full overlap integral half, and then if they go too far, the molecules in this cell, they are coming closer, complete overlap. If they are going far, you have a very less overlap integral. So essentially we are doing experiment in multidimensional to look at the fluctuation that is happening, and we are understanding the diffusion, 2-dimensional, 3-dimensional diffusion and all those, and we can plot it correlation function to look at the fluctuation of the fluorescence that is happening, right. So various kind of autocorrelation functions demonstrate the enormous difference in the mobility of buffer in solution. In cytosol, you can have a molecular motion that is happening in buffer, in cytosol, in membrane with some receptor, and you can understand inside the cell what is happening. So from a pure solution that I started to show you for doing fluorescence, now we are dreaming to do inside the cell, inside the brain tissue, how the serotonin, dopamine and all those are fluctuating, how the molecule is binding to other molecule. So that is the power of a fluorescence spectroscopy, that it helps you in dreaming, going from a pure protein, purified protein, to the complex cell or tissue level to understand what is happening in a real life, right. So if you look at the membrane motion is very different than the buffer or even the cytosol. So complexity keeps increasing as you as you go to the real scenario, but biophysics often start with a simpler one, and then you increase complexity to go it, otherwise you do not know what is happening. That is how we build up step by step to understand the phenomena, biological phenomena that is happening. Start with a simpler system, then go to complex and complex and complex, right. So let us conclude here. Fluorescence lifetime we can use to measure dynamics. Fluorescence lifetime can be measured either in the frequency domain or the time domain, right. Some of those time domain data I showed you. Time correlated single photon counting, that is TCS-PC, a common technique to measure the fluorescence decay in the time domain. FRET that we discussed is a spectroscopic ruler for a distance dependent phenomena, how they open and close, and molecular mechanism you can give rise to fluorescence fluctuation, you can understand the particle movement, conformational change, chemical and photochemical reactions that could be happening. So fluorescence has a real application in many of the biological experiment that we do. Here in IIT Bombay we have a confocal microscopy, you can see in the cell we have G stack, right. You can take a slice of your tissue and cell, you can do. So lots of this room, next room is your fluorescence based facility that we have. Confocal and spinning disc we have in the, and then first year when you go for a practical you have a spectrophlorimeter to start with. But that is the length and breadth of fluorescence spectroscopy and its application in life science research. Developmental biology ho, neuroscience ho, biophysics ho, biochemistry, everywhere it has a profound application, and lots of applications are still developing in the real world that we discuss some of those, but even beyond that lots of things are developing to use fluorescence in life science research. Okay, let us close it here for fluorescence. Thank you.